Hello friends, good morning. Welcome to UDAAN Career Institute once again. In today's class, we will continue with chapter 2, that is reproduction in flowering plants or reproduction in angiospermic plants. In the last class, we have started with the introductory part. We have gone with the flower in detail. Also, we have covered pre-fertilization structure and events. In the last class, we started with a male reproductive organ, that is androecium, or it is also known as microsporophyll. So in today's class, we will continue with the same topic, that is male reproductive organ. Now friends, as I have told in the last class that stamen is considered as male reproductive organ of a flower. Now, if you see the structure of stamen, it consists of two things. You can see here that there is a presence of anther and there is a slender, long stalk-like structure which is known as filament. So, stamen is composed of anther and filament. If you see the cut section of the anther, there is a three-dimensional cut section of the anther which has been shown here, in which you can see there are chambers which are present in the anther, and within those chambers the pollen grains are present, and there is a line of dehiscence which you can see, which leads to the release of pollen grains. Now going to into more detail regarding the male reproductive organ. Here, as the figure shows, that there are two parts of a stamen. Stamen consists of two parts. The first one is a long slender stalk-like structure which is known as filament, and the terminal bilobed structure which is known as anther. So anther is a bilobed structure. If you see the structure of anther, you can see presence of two different lobes. The first lobe and back side there is the another lobe. So basically anther is a bilobe structure. So there are two parts of stamen, a long slender stalk-like structure known as filament, and an anther which is a bilobe structure. Now the proximal end of the filament, it is attached to the thalamus or the petal of the flower. Now if you see the structure of anther, this part of the diagram is considered to be the proximal end of the filament. Now this proximal end of the filament, it is either connected to the thalamus or to the petal of the flower. Now the number and the length of the stamen are different in case of different flowers. As you can see that there are wide variety of flowers. In those flowers, the length of the stamen and the number of the stamen also differs to a great extent. Now if you collect stamen of different flowers, from 10 different flowers, if you try to collect the stamens, in each of them you can see that there is a wide difference in their size, in their length, as well as you can see in their numbers also. So on careful observation of stamen under a microscope, we can see a neat diagram on the basis of which we can elucidate there are different kind of shape and attachment of anther in different flowers. Now a typical anther is bilobed in structure, means it has two lobes. As we have gone earlier, that anther consists of two lobes. Now each lobe has two theca, that is they are dithecous. Now if you see the anther in detail, in that case you can see that basically each lobe of anther, it consists of two pollen sacs or two microsporangia. So each lobe, it has two theca, and these theca will be having the pollen sac, or these theca will be having the microsporangia. That is, each lobe has two theca and they are dithecous. So totally there will be four theca in two lobes of anther. Now there is a longitudinal groove which runs lengthwise and separates both the theca. Now if you try to understand different types of tissue and their organization in the transverse section of anther, we can see that there is a bilobed nature of anther. Anther is bilobed. There are two lobes in the anther, and it can be clearly visible in the transverse section of the anther. Now if you try to have a transverse section of an anther, then both the lobes of the anther are clearly visible. Now anther is a four-sided tetragonal structure. Now as you can see the transverse section of the anther, if you see the transverse section of the anther, here you can see that there are four different areas or four different sides. We can see there are four different sides. It is a tetragonal structure and it consists of four microsporangia, and those microsporangias, they are located at the corners, two in each lobe. So if you see the structure, there are two microsporangia in each lobe. So there are two different lobes and each lobe has two microsporangia, or as we have gone earlier, that each lobe has two theca, that is they are dithecous. The microsporangia, they further develop and they will become the pollen sacs. So within each lobe there will be two pollen sacs. They extend longitudinally throughout the entire length of the anther. So the microsporangia or the pollen grains will be present throughout the length of the anther, and these microsporangia or pollen grains, they will be packed with the pollen grains. So pollen sacs will be containing those pollen grains. The male reproductive organ is also known as androcium and their unit is known as stamen. Now starting with the structure of microsporangium. Now friends, as we have gone earlier, that anther is bilobed. Each lobe has two theca, that is they are dithecous, and within each lobe of the anther, two microsporangia will be present at the corner. So totally there will be four different microsporangias, and those microsporangia, they will mature and they will get converted into pollen sacs, and those pollen sacs will be containing pollen grains. Now we have to understand the structure of the microsporangia. That is, what is the exact structure of microsporangia. Now, if you see the transverse section, each microsporangia, it is circular in outline. So if you see the section, if you see the diagram A, you can see that the microsporangia, it is circular in nature. So the structure B, it is an enlarged view of a microsporangia where you can see a circular outline of the microsporangia. You can see it is circular in nature. Now, if you see the microsporangial, the structure of the microsporangium, it consists of four layers. There are four different layers which are present in microsporangia. So if you see from outside towards the inside, the outermost layer is the epidermis, which is followed by endothecium. Then there are 1 to 3 layers of middle layer, and the innermost layer is known as tapetum. Once again I'll repeat, microsporangia has four layers. If you see the transverse section of microsporangia, there are four different layers which are found here. Outermost is epidermis, followed by endothecium. Then there will be 1 to 3 middle layers. So middle layers may vary from 1 layer up to 3 layers. So from 1 to 3 layers, it will be known as middle layers, and the innermost layer is known as tapetum. So out of 3 layers, that is epidermis, endothecium and middle layers, they perform the function of protection, and it also helps in the dehiscence of the anther for the release of pollen. So here friends, if you see the structure, that is the diagram C, you can see that the anther has been dehiscence, means it has been broken, the outer wall has been broken, and it leads to release of the pollen. This is known as dehiscence of anther. So the outer 3 layers, they perform the function of protection and dehiscence of anther for the release of pollen, and the innermost wall is or the innermost layer is known as tapetum. tapetum, the function of tapetum is nourishment of the developing pollen grains. It provides nutrition to the developing pollen grains. Also, it secretes hormones and enzymes, and also it forms sporopollenin. Now, if you see sporopollenin, it is considered as one of the most resistant substance which is found nowadays. So sporopollenin, it is resistant to any kind of enzyme, or sporopollenin is also resistant to acids. Sporopollenin is resistant to strong alkalis. As a result, due to the presence of sporopollenin, pollen grains can be preserved as fossils. So basically it is the tapetum which is secreting hormones, enzymes and sporopollenin which forms the protective part of the anther. The cells of the tapetum has dense cytoplasm, and generally they have more than one nuclei. So basically the tapetum cells, they have dense cytoplasm and more than one nuclei. They secrete hormone and enzymes. They also form sporopollenin, and this sporopollenin will increase the thickness of the exine. Now, there will be free nuclear division which will be taking place in case of tapetal cells, and which is not followed by cytokinesis, and this is the reason that tapetum, it has more than one nucleus. So only nuclear division will be taking place and no cytokinesis would be taking place in tapetal cells, resulting in formation of more than one nucleus in the tapetal cells. Now basically when anther is young, then initially a group of compactly arranged cells which are known as sporogenous tissue, it will occupy the center of microsporangia. So initially, during the early stage of development of anther, the microsporangia will be filled up by sporogenous tissue, and later it develops into pollen sac. So basically, if you see the structure of the microsporangia here, we can see that the microsporangia are circular in outline. They have 4 different layers or walls, we can say, starting from epidermis, endothecium, middle layers, and the tapetum. From outside towards the inside, there is a line of dehiscence where we can see there will be release of pollen grains coming out of the pollen sac. Now coming to the another part, that is the microsporogenesis. Genesis means formation of microspores or formation of pollen grains, basically is known as microsporogenesis. Now as we have gone earlier with the structure of microsporangia, now we'll move towards microsporogenesis. As the anther develops, the sporogenous tissue, the cells of sporogenous tissue will undergo meiotic division, and as they are undergoing meiotic division, it leads to the formation of microspore tetrads. So there will be 4 different microspores which will be formed, and they are known as tetrad because initially all 4 microspores are attached to each other. Now the ploidy of the cells of the tetrad will be haploid. Sporogenous tissue, the cells of sporogenous tissues are diploid in structure, they are diploid, and they undergo meiosis. Once they undergo meiosis, it results into the formation of 4 microspores. All 4 microspores will be haploid because of the meiotic division which has taken place. As each cell of sporogenous tissue can give rise to a microspore tetrad, each one is considered as a potential pollen, or we can say microspore mother cell. So every particular cell of sporogenous tissue, it can be considered as microspore mother cell because it will undergo meiosis and it will lead to the formation of microspore tetrad. That's why they are considered as potential pollens. This process of formation of microspores from a pollen mother cell through the process of meiosis is known as microsporogenesis. So as I have told earlier that initially these microspores, they are arranged in the cluster of four cells together. That's why they are known as tetrad. So after meiosis which has taken place, initially all four microspores will be attached with each other. Now, as the anther undergoes maturation and dehydration, all these microspores, they will dissociate from each other and they will live as an individual pollen grain, or they will develop as an individual pollen grain. Inside each microsporangium, we can see thousands of microspores or pollen grains are formed, and when the anther descends, all these pollen grains are released outside. So here you can see that there is formation of a microspore tetrad from a microspore mother cell, which we can say that it is a cell of sporogenous tissue. Through meiosis 1 and through meiosis 2, four different microspores have been formed, all together attached with each other in the form of a tetrad. So friends, once again we'll briefly go through the points. In today's class we have gone with male reproductive organ of a flowering plant, which is the stamen. Stamen consists of two parts, that is a bilobed anther and a filament. Filament is a long slender stalk like structure, and the anther has two lobes. Now each lobe has two different theca, so basically it is known as dithecous. Basically they are dithecous in nature, so totally there will be four theca. Now microsporangias are located in the corners in each particular lobe, so entire anther consists of four microsporangia. During the initial stages of the development, it is known as microsporangia, which further develops into pollen sac. Now, if you see the structure of microsporangia, there are four different walls of microsporangia and it is circular in outline. Now, the four walls are epidermis, endothelium, middle layers, and tapetum from outside to inside. The first three layers from outside to inside, that is epidermis, endothelium, and middle layers, will perform the function of protection and will help in the dehiscence of anther. The innermost layer of the microsporangia, that is tapetum, it basically provides nourishment. It also secretes hormones and enzymes, and it also leads to the formation of sporopollenin. Now tapetum has dense cytoplasm and have more than one nucleus. The cells of the tapetum, they are basically binucleated cells, means they have more than one nuclei because of free nuclear division taking place. Now microsporogenesis means how microspores are formed. This involves the process of meiosis. Sporogenous tissues, they are present in the microsporangia. Each sporogenous tissue, the cell of each sporogenous tissue, it will behave as microspore mother cell, which will undergo meiosis 1 and meiosis 2, leading to the formation of four microspores. All four microspores are attached together in the form of tetrad. As the anther undergoes maturation, these microspores will get segregated from each other, they will get separated from each other, and they will develop in the form of an individual pollen grain, which gets liberated after the dehiscence of anther. So friends, in the next class onwards, we will start with the structure of pollen grain, which is the male gametophyte. Thank you very much.